Good afternoon. I'm going to talk about behavioral epigenetics, which is a very recently established discipline that can, in general terms, be defined as the field of research that tries to elucidate to what extent epigenetic mechanisms influence and determine behavior in animals. And I'm going to talk more specifically about mammals and more specifically the humans. A very brief introduction to epigenetics. I'm sure everybody knows what epigenetics is. It was Conrad Waddington that in 1942, in his paper, The Epigenotype, wrote, "We certainly need to remember that between genotype and phenotype and connecting them to each other lies a whole complex of developmental process." He introduced the term epigenotype to therefore define the set of developmental processes linking the genotype with the phenotype. The use of the word epigenetics has suffered some changes over the last decades, but its current stricto sensu version primarily refers to the field of research that studies the group of cellular mechanisms that participate in the regulation of gene expression, thereby resulting in their silence or activation. This complex regulation system operates without changing the nucleotide sequence of the underlying DNA it acts upon. These changes are mitotically and meiotically heritable, so heritable from somatic to daughter cells in the first case, and from gametes to offspring in the meiotically case. But at the same time, they apparently demonstrate a plasticity that could be used, and we will see this later, in therapeutic settings. So, okay, this set of cellular mechanisms that participate in the regulation of gene expression, activating, silencing the genes, what are these epigenetic mechanisms? The three most important epigenetic mechanisms are currently thought to be the methylation of certain DNA bases in the DNA strand. And here I show the action of these enzymes, the DNMTs. You can see in the first image the DNA strand, a CpG site, cytosine guanine site. This is the enzyme, the DNMT. This is the methyl donor, S-adenylmethionine. This is the methyl group, and this is the result. The first DNA strand does not have these methyl groups. The last one, as you can see, is already methylated. This will result in the suppression of this particular gene transcription. This is the first of the three most important epigenetic mechanisms known to date. The second one is a variety of modifications that take place in the chromatin proteins, the histones. And these modifications are especially acetylation, methylation, but also ubiquination and phosphorylation. This is a DNA strand. Those ME groups we have already seen in the first slide. This is the folding of the chain around histones. These are the histone protein tails, and I'm going to show you in the next slide. Here, this is a detail of the histone with its tail, a set of modifications that can occur, and it constitutes the second epigenetic mechanism. Acetylation here. This is an amino acid of the protein tail. Ubiquination, the phosphorylation, the acetylation, and here this is the first mechanism we saw in the first slide. The third mechanism is the microRNAs, associated post-transcriptional gene silencing, that end up with the degradation of target messenger RNAs and the translational inhibition of target mRNAs. So, in a very recent study from 2012, Makani and colleagues, the study was called, Cigarette Smoke Exposure Associated Alterations to Non-Coding RNA. So, this microRNAs is one of the three most important non-coding RNAs, and they investigated how the cigarette smoke exposure was associated to this kind of alterations, and it is in fact associated. So I'm now introducing a third element, which is the environment, and the relationship of the environment with these epigenetic mechanisms. In this study, they write, follow-up analyses show that several of these transcripts exhibit increased expression in many lung cancer and breast cancer cell lines. So we are seeing environment, cigarette smoke, epigenetic mechanisms, specifically this one, and phenotype, in this case illnesses, cancer and breast, lung cancer and breast cancer. So, these three epigenetic mechanisms are thought not to act in isolation, as you may already, of course, conclude, but they are thought to participate in a complex web of interactions and feedbacks where the genome itself, of course, and environmental factors like cigarette smoke, in this example, also step in. Let's talk about a little bit about the environmental factors. Although it can be difficult to ascertain whether it are genes or the environment that have a bigger impact in the development of human disease, traditional epidemiological thinking asserted that disease is either caused by genetic or environmental factors. However, one of the most remarkable features of the epigenetic mechanisms is that they appear to connect the environment and the genome, like we've seen in the previous example. And in fact, like Kubota writes in 2012, in medical research, it is now believed that environmental exposures interact with the epigenome and influence the possible onset of some diseases. More than recognizing that this new factor, the environment, has entered the physiopathological and epidemiological equations, it looks like as if the molecules that regulate gene expression, these epigenetic mechanisms, could be modulated by environmental factors, that is to say, by nurture. I've mentioned the cigarette smoke, which can be considered toxin, but also nutrients and psychosocial interactions. The main focus of my presentation will focus specifically in this third kind of environmental factors, but several experiments have been done with the influence of toxins and nutrients. Randy Jertel's experiments with either BPA, which is a toxic product that is present in plastic bisphenol, and with nutrients like folic acid that is given to pregnant women, have also come to the same results. So, the epigenetic mechanisms are already known in the human development. For example, the case of the X chromosome inactivation, which occurs in all mammal females, and in humans, in some diseases, like in cancer. Moshe Schiff says that when genes that typically suppress tumors are methylated, so are methylated by these epigenetic mechanisms, the tumors metastasize. But unlike genetic mutations, epigenetic changes are potentially reversible, and this is one of the major key points and interest around epigenetics. In fact, a mutated gene is unlikely to mutate back to normal. The only recourse is to kill or cut out all the cells carrying the defective code. But a gene with a defective methylation pattern might very well be encouraged to reestablish a healthy pattern and continue to function. So this is the principle that underlies the use of some drugs to treat already some cancer diseases. Given the epigenetic effects of the DNMT enzymes that I've showed you some slides ago, some DNMT inhibitors are approved or are under investigation for treatment of some cancers, like azacitidine for myelodysplastic syndrome, or for acute myeloid leukemia, or decitabine for acute myeloid leukemia and chronic myeloid leukemia. Okay, let's, before going to behavior, in physical disease, we also have the case of multiple sclerosis. Until just 3 or 4 years ago, nobody thought that epigenetical mechanisms could be associated with this very disabling disease. But Koch and colleagues in this year's article, they write that multiple sclerosis is a disease, well, everybody knows, of the CNS, the central nervous system, with both inflammatory and neurodegenerative features. All those studies on epigenetic changes in multiple sclerosis only began in the past decade. A growing body of literature suggests that epigenetic changes may be involved in the development of this disease, possibly by mediating the effects of environmental risk factors such as smoking, vitamin D deficiency, and Epstein-Barr virus infection. So these are some examples of the importance of these, of the study of epigenetic mechanisms in human disease, but I'm going now into behavior. One of the landmark papers in behavioral epigenetics was written by Weaver, Schiff, Mini, and colleagues, and published in 2004 in Nature Neuroscience. In this study, they show that an epigenomic state of a gene can be established through behavioral programming, and it is potentially reversible. The study suggested a causal relation among epigenomic state, glucocorticoid receptor expression, and the maternal effect on stress responses in the offspring. That is to say, an influence in a very concrete behavioral feature. I'm now going to explain this in a... So what does all this mean? That some epigenetic changes can occur after birth through the mother's behavior towards her offspring. So the study, they basically studied the effect of the mother's nurturing. They measured the leaking and grooming activity of the mothers to the offspring. And this leaking, they concluded that this leaking grooming activity triggered a cascade of events with the release of serotonin, which triggered an epigenetic difference in the hippocampus cells of the newborn, which was the first mechanism, DNA methylation, which triggered a different glucocorticoid gene receptor expression, which altered the HPA axis balance, the hypothalamus pituitary adrenal glands axis, which resulted in different responses to stress of the offspring. So there were two groups. The mothers that leaked and groomed more, they saw that there was a methylation change that led to less glucocorticoid receptor gene expression, that led to the production of less cortisol, and the effect was more calm pups. The mothers that leaked and groomed less, the result was completely different, more nervous and fearful, avoiding pups. but they also show that the dysmethylation that occurred is potentially reversible. Unlike altered genes, which are disabled for good, methylated genes can be demethylated, and vice versa. Methyl marks can be regained via nutrients, drugs, and enriching experience. In this particular case, Schiff and Mini, for instance, Schiff was able to remethylate the glucocorticoid receptor gene with the administration of methionine, an essential amino acid that contains methyl, to well-nurtured rodents. Mini reversed epigenetic markers by injecting a drug in the brains of adult rats. And this drug is chemically similar to the histone deacetylase inhibitor valproic acid, which is used clinically in people as a mood stabilizer and which increases the effectiveness of antipsychotic medications in the treatment of schizophrenic or severely depressed patients. So, the behavior of the mother affects the gene expression and some part of the brain circuitry development of the offspring. Going back to the first presentation of this scheme, now instead of environment, let's put here the behavior, the mother, but also possibly the family and other socialization factors that influence the epigenome that leads to changes in the phenotype. And in humans, it is much more difficult to study this in humans. Because you cannot make samples from human brains. Some post-mortem studies have been made, have been attempted. Well, another line of research has tried to compare the rise of epigenetic differences in people with identical genomes. The comparison of monozygotic twins' epigenetic states showed that these similarities not only exist, but that they grow over life, especially in the twin pairs that experienced different environmental living conditions. As Randy Jertle claims, although the epigenomes are similar in young twins, they are increasingly different in older twins, especially if the twins have lived in different environments and have had different social habits. Interestingly, identical twins can also vary in their susceptibilities to disease and their physical and behavioral characteristics. Of course, Jertle notes that although there are several possible explanations for these observed differences, this is not the one possible explanation. One is, in fact, the existence of differing epigenomes. So even if it is still not clear to what extent these epigenetic differences are responsible for the phenotypical dissimilarities displayed later in life, as Murgatroyd and Spengler say, understanding how early life experiences can give rise to lasting epigenetic marks conferring increased risk for mental disorders, how they are maintained and how they could be reversed is increasingly becoming a focus of modern psychiatry and should pave new guidelines for timely therapeutic interventions. In conclusion, the research on the influence of epigenetic changes in behavior is still in its infancy, I would say in the womb. And broad, comprehensive and well-designed studies of epigenetic mechanisms in human patients with behavioral pathology are scarce. However, there's already a growing, however little it's growing, body of evidence that shows that some molecular mechanisms underlying behavior are epigenetically regulated. In biomedical terms, it makes us put an emphasis on prevention more than on therapeutical approaches. And epistemologically, we can ask, so is there here a role of nurture on nature, an influence of nurture on nature? And what can developmental psychology say to biology? because behavior is having an influence on the gene expression, we have here an interesting relationship between these two sciences. That's it.